orient biologically here, right? So really the essential functions of a synaptic connection are to release vesicles to signal, and then to utilize endocytosis to recover the cargo's membrane and machinery to allow for subsequent rounds of release. And so these two domains of exocytosis and endocytosis are, conventionally thought to be spatially segregated at the synapse into an active zone where the exocytic machinery is tethered, and the endocytic periactive zone, which is generally thought to surround it, where endocytosis happens. Although I think this domain is particularly poorly defined in terms of its molecular identity and dynamics and function. And so for the talk, I'm really focusing here on the presynaptic neuron and presynaptic vesicle endocytosis. And so just very generally speaking, we know a decent amount about presynaptic compensatory endocytosis. We know, for instance, that it can occur very robustly and adaptably to a wide range of exocytic scales, so and over very different time scales. So at single action potentials, at physiological temperatures, endocytosis can occur extremely rapidly by ultrafast endocytosis, which can occur within hundreds of milliseconds and can regenerate 1 to 3 vesicles. Under normal to low moderate activity levels, vesicles can be regenerated by clathrin-mediated endocytosis, which can happen on a time scale of tens of seconds. And then as stimuli increase and vesicle rate and exocytosis begins to increase, additional mechanisms can be engaged, such as bulk endocytosis, which can take up large tracts of membrane and then subsequent vesicle regeneration from those large membrane cisternae. And so the key idea here is simply that that endocytosis at synapses is very adaptable, both with regard to its scale and also its speed and latency. And so what do we know about endocytosis? So we know that different synaptic architectures in different organisms with different vesicle pool sizes and firing properties are all fundamentally utilizing similar and conserved mechanisms of vesicle endocytosis and recycling. And we know this from assays like capacitance measurements that allow very highly time-resolved measurements of membrane dynamics, from EM studies, for decades, which have provided very high degree of spatial detail in terms of the membrane remodeling dynamics, assays that can track endocytosis kinetically, such as dye uptake and release, or pH-sensitive fluorescent protein dynamics. And then, very rare cases where there's single molecule or single site insight into the process. And so I think, despite all these many approaches and many decades of work, I think there's one area, to my mind, that is still glaringly unknown, and that's that we don't really have any time-resolved molecular dynamics that drive these processes of membrane internalization. I'll get into why that's a limitation in a minute. But what the machinery that I'm interested in, the machinery that I want to know, these time-resolved dynamics, the main function of endocytic machinery is to generate actin-dependent forces, or forces generally, at membranes to deform the membrane and drive endocytosis. And this happens via the concerted activities of dozens of proteins that interface with the plasma membrane, with adapters, and with proteins that regulate actin dynamics and assembly. And so the key features here are that these different modules between the membrane, the adapters, and the actin, they're all densely interconnected, both between modules and also within modules. So you have both many proteins and a very high degree of interconnectivity between them. And this is true for synaptic endocytosis proteins and also in non-neuronal cells. It's the same machinery. And so what's so interesting about studying this machinery at synapses? So I would say, so the most obvious difference is simply the way these molecules are distributed at synaptic membrane. So on the left here is a model synapse. This is the fly neuromuscular junction, which is the model system that I work on. And on the right here is a HeLa cell. And so this is in magenta, the fly protein nervous rec, which I'll tell you about in more detail in a minute. And then it's mammalian homolog FCHSD2. And you can see quite clearly that at the synapse, this protein is decorating this large micron scale sort of mesh-like reticulum, whereas in non-neuronal cells, it's accumulating in these spots, which are effectively the size of a site of endocytosis. And these white dots here are meant to indicate the size of a site of clathrin-mediated endocytosis in each of these images. And so that's really one glaring difference, but also there's other differences. So in addition, at synapses, and this is true not of simply the fly neuromuscular junction, but other synapses, and this has been measured both by imaging and by proteomics, endocytic machinery accumulates at very high concentrations at synapses, so over 10 micromolar, almost universally. And then this may contribute to its very broad membrane distribution. But it's really completely unknown what the dynamics of these proteins are at these synaptic membrane domains. By contrast, in non-synapses and non-neurons, the same proteins are accumulating at many-fold lower concentrations, so typically less than 1 micromolar. And they have this very stereotyped nanoscale distribution and a very stereotyped pattern sequence of recruitment. And so in contrast to this well understood dynamics here, we really just simply don't know what are the fundamental rules that are governing this large scale organization, dynamics, and activity of the endocytic synaptic membrane cytoskeleton remodeling machinery. And again, this is in contrast to the same proteins in non-neuronal cells. So this is just an example of these proteins. So in magenta and green, 2 different endocytic proteins. And what you can see is, again, that they accumulate in these discrete puncta. And this has really been important for the field to understand how these molecules are recruited and activated, because using methods like TURF and live imaging of yeast, the field has been able to really get a very high spatial and temporal resolution of their pattern of recruitment. And that's led to a model where there's a very stereotyped canonical sequence where there's an early recruitment of adapter proteins that can bind to cargos and membrane. The endocytic site matures as actin assembles and deforms the membrane. And then finally, the membrane undergoes fission as dynamin constricts the neck and the vesicle buds off. So while these morphological processes are recapitulated at the synapse, it's not clear to what extent the same molecular dynamics are occurring. And so I would suggest that the previous image raises the possibility of whether these proteins are in fact pre-deployed to the membrane, and whether these subsequent remodeling steps represent different stages of activation of these proteins. And so suggesting there might be a completely different regulatory logic to their activity at synapses. And so the types of questions that I'm interested in are, how do these dozens of endocytic adapters and regulators function collectively? So most work is really focused on individual mutant phenotypes or pairwise regulatory interactions. Are the mechanisms that regulate the molecular dynamics and activation of these proteins the same at synapses and as at non-neuronal cells? And then ultimately, how do synapses control the recruitment versus the activation of this endocytic machinery? And so why is it important to consider the molecular dynamics? Why isn't it enough to simply look at a mutant phenotype and identify, all the genes that are required? So I would say that, there's a lot of things that we can learn really uniquely by studying the molecular dynamics. So where and when do different events occur relative to sites of synaptic vesicle release? So we know, for instance, that at the NMJ, different sites of release can have up to 50-fold differences in their inherent release probabilities. We could ask questions like, how does endocytosis scale with exocytosis? Is it simply the frequency or the scale of the events, or both? And how does the molecular machinery adapt to these scaling rates of exocytosis? And we know that different stimulation paradigms result in different rates and kinds of endocytosis. And finally, what are the mechanisms that confer this adaptability and robustness to vesicle recycling? And one of the challenges, I think, to understanding this is that lots of mutant phenotypes really have very intermediate or adaptive phenotypes. And so that makes it hard to really unambiguously ascribe specific functions to individual genes, simply by looking at the effect on endocytosis. And so what I'm going to tell you today is sort of two stories. One about what I've learned about the activation of the endocytic machinery using a module of proteins. And then in a second part, I'll tell you some more preliminary work about some studies I'm doing on how these molecules are organized in this micron scale mesh at the synapse. Okay, so I'm going to just start by describing this in vitro model system and the proteins that I'm studying. And so again, the main function of these proteins is to tether actin assembly and force generation to the membrane. And so I'm using a model here of two proteins. One is nervous rec, which is the fly homolog of FCHSD2. And that directly binds to membranes via its bar domain. And then through its SH3 domains can interact with WASp, a protein which drives ARP2/3 mediated actin assembly. And so together, the idea is that this is a representative module that couples actin assembly and membrane binding. And one of the interesting features about this protein module is that it's highly regulated by autoinhibition. So these SH3 domains of nervous rec can fold under the bar domain, and so block both the barred membrane interactions and also the SH3 protein interactions. And one of the unique features about this protein in particular is that it remains autoinhibited, at least partially, even when it's on membranes. And this is actually really unique among these barred SH3 proteins. Typically, when one of them binds to the membrane, the SH3 domain is just free to flop around and interact with its binding partners. Whereas with nervous rec, previous grad student found that the SH3 domain stays tucked in against the bar domain, even when it's on the membrane. And that's true even when it's bound to WASp. And so the idea is that nervous rec on its own is actually a very poor activator of WASp-mediated actin assembly. And so that made us consider the hypothesis that maybe some additional interactions via this second SH3 domain might be important in controlling the activity of this protein module. And so the candidate that we examined is this protein DAP160, which is the fly homolog of intersectin. And it interacts with the SH3 domain of nervous rec, the proline-rich domain of WASp, and it has shared genetic phenotypes with both of these proteins. And so I'm just going to highlight a couple of the biochemical studies that we did that I think illustrate the main function of this collective module. So this is a pyrene actin assembly assay. So basically, the fluorescence increases over time as actin assembly increases. And what we find is that actin alone doesn't assemble very well because the nucleation limited. Adding WASp doesn't really do much because itself is autoinhibited. Adding nervous rec to this mixture has only a very incremental effect, as does adding DAP160. But then combining all of the proteins together with lipids, so with synthetic membranes, we get finally this robust activation. And so the take-home message from this is that robust actin assembly requires the collective binding of all three of the proteins and the membranes. And so then we looked at the membrane binding directly using a liposome co-sedimentation assay. So we just take purified protein and membrane, mix them together and spin it down and see what's stuck to the membrane. And what you can see is that nervous rec alone binds membranes fairly poorly because it's autoinhibited. Adding DAP160 enhances membrane binding, but adding DAP160 and WASp increases binding. This is actually to a maximal level. So this is the same degree of binding as the uninhibited bar domain alone. So suggesting that the proteins, when they're all together, they're driving maximal membrane recruitment. And so then we can visualize this directly. So this is just one more in vitro experiment, taking the proteins in an aqueous buffer, and this is floating in a sea of lipid. And so there's a membrane lipid interface here right at the boundary. And then we can ask what happens to these different nervous rec variants. And so if we take normal nervous rec, which is autoinhibited, and we mix it together with WASp and DAP160 and this green actin, we see that membrane actin assembly occurs, and it's preferentially tethered to this lipid interface here, highlighted here in green, and then you can see nervous rec decorating the membrane, the lipid interface. If we take a variant of nervous rec that is deinhibited and mix it in the same conditions, we see now that actin assembly still occurs, but now it's become decoupled to the lipid interface. So instead now you see these punctate nervous rec asters that are nucleating the assembly of actin filaments that aren't in contact with the lipid interface. And so this suggests that what autoinhibition is doing, in the combined interactions of these, or it's really constraining actin assembly to the lipid interface. So it's really requiring that it is associated with the lipid. And so just to summarize that in vitro portion, so again, the model is that nervous rec, this membrane binding protein, is autoinhibited on the membrane. Even when it interacts with WASp, it's still autoinhibited. And so we're considering this to be a clamped state where actin assembly isn't occurring despite the presence of these regulators. And then plus DAP160 plus the membrane, we do see robust membrane-coupled actin assembly. And so suggesting that activation really reflects this strong coincidence detection among all of these components. And so then the question is, how do these mechanisms actually control synaptic actin assembly? And so the model system that we're using here is the fly neuromuscular junction. So we take a late third instar larva, slice it down the middle, and unfold it. And then we're looking at the motor neurons that innervate these crawling muscles. And so this is the entire synaptic contact for this neuromuscular junction. And then for the rest of the talk, I'm really just going to be zooming in on one or two boutons, which is at this level of scale. And these boutons have up to tens of individual sites of synaptic vesicle release called active zones here, labeled in cyan, and then again this very abundant and broadly distributed endocytic meshwork here, labeled in magenta and green. And so again, so the idea is that this very broad distribution, while it tells us that they're all at the membrane, it's really impossible just by looking to tell where these proteins are active. And so the strategy that we took to answer this question was to look at actin assembly. And so on the left here is a time lapse of actin assembly in budding yeast, and these spots here are all endocytic associated actin assembly. And then when we compare that to actin assembly at the NMJ, we see a really similar morphological dynamic pattern of actin assembly. And you can tell that this isn't this broad reticular mesh of actin, it's assembling in these dynamic puncta. And so the reason that I think this is a great model to understand how these are activated is because we know that actin dynamics are required both at this synapse and other synapses for synaptic endocytosis. And this is really a fundamental direct readout of the activity of these proteins. And so to quantify these patch dynamics, I used a tool developed by Julien Barraud at Yale, who studies yeast actin assembly in endocytosis. And so we use this to identify sites of assembly, track their intensity dynamics over time, and then from that we can extract their frequency, their duration, and their overall dynamics. And what we find is that at the NMJ, the average lifetime of these patches is quite similar to that in yeast, only much broader. We see that they occur with a frequency which approaches the expected rate of exocytosis at the synapse. And I should note that we're doing these experiments under conditions where the only exocytosis is spontaneous release. We're not stimulating. And so just to generally characterize these actin patches, so we think they're likely sites of endocytosis based on their morphology and duration and frequency. And we see that they frequently co-label with the endocytic adapter AP2. So this is an F-actin marker here in magenta and the endocytic adapter AP2 in green. And you can see these areas of overlap that's quantified here. And then when we disrupt endocytosis using a temperature-sensitive Shabiri mutation, so this is a mutation in dynamin, which effectively shuts down vesicle cycling in this synapse, we see a significant decrease in actin patch formation, so suggesting that it is coupled to endocytosis. And then asking about the direct molecular determinants of the actin assembly. So I looked at this WASp ARP23 module that I've described. So again, just a reminder, so WASp promotes, activates ARP23 to drive branched actin assembly. We see really striking co-localization between our F-actin marker here in magenta and ARP3 in green. That's just quantified here. And then when we look in a WASp mutant, we see a significant decrease in the frequency of actin assembly. So suggesting that, again, that this machinery is required for the formation of at least some of these. And I should note that this is a hypomorph, so there could be still some residual activity. And so then the question is, how does this clamping, where nervous rec is a really poor activator of WASp on its own, versus the collective activity, how does this play out in vivo? How does it actually control these dynamics? And so you can imagine a couple different outcomes. So if the majority of nervous rec at the synapse is acting as an activator, then we might expect to see either fewer, briefer, or sort of wimpier actin patches. Whereas if the majority of this pre-deployed nervous rec is acting as a clamp on actin assembly, then we might expect to see actually more actin assembly. And I think this clamp model is particularly important to consider in light of the fact that there's many, many other proteins that could be activating WASp either concurrently or in response to a loss of nervous rec. And so when we look at this actin assembly in a nervous rec mutant, it turns out that we see an increase in the frequency of patch assembly. So this is control here and nervous rec null mutants. And we see this significant increase in the frequency of actin patches. When we measure the distribution of how long these patches last, we actually don't see any change in their duration. So it's really just a pure increase in the number of assembly events that are occurring. And we see the same phenotype in a DAP160 null mutant. And we also see, perhaps more convincingly, the same mutant phenotype when we simply remove the interaction domain between DAP160 and nervous rec. So this is a DAP160 null mutant in which we express specifically in the motor neuron either a full-length DAP160 or this DAP160 which is missing this interaction domain. And again, we see the same phenotype of more actin patches, but no change in sort of the properties of the ones that are left over, in the dynamic properties of the ones that are left over. And so then the question is, what are all these extra patches? Is this more endocytosis happening? Is this just random actin assembly? And I think from our in vitro experiments, we would really expect that this would be kind of uncoupled from the plasma membrane because we're losing this coordinated autoinhibition. And so to test that, I looked at membrane uptake and recycling at the synapse using FM dye labeling. So I just stimulated exocytosis and loaded FM dye, and then asked how much dye got loaded as a measure of internalization. And so what you see is that with the full-length rescues, this is the same experimental conditions as the previous experiment. We see normal uptake. We see significantly lower membrane uptake with our domain mutant that blocks interaction between DAP160 and nervous rec. And then as a positive control, we see really complete lack of internalization in this Shabiri dynamin mutant, the same one that blocks, interrupts the actin assembly in that first experiment. So suggesting that that this module is both constraining actin assembly, but really directing it also to functional events that are contributing to endocytosis. And so the summary model here for this first part is that so nervous rec is autoinhibited, and then it's autoinhibited even when it's complexed either with DAP160 or WASp. And so this would be a mechanism to explain how you could get this really broad pre-deployment of these proteins to the membrane without necessarily driving actin assembly everywhere. And that it really requires this full relief of autoinhibition, either by the coincidence detection of all of them, or maybe some other synaptic cues like membrane charge or cargo. And that's driving really productive membrane-coupled actin assembly. So that's part one. I can pause here if people have questions on this part. And this is all also available online as a preprint. So you can feel free to check that out. Otherwise, I can plow ahead and tell you about this really broad meshwork of proteins at the synapse.
Stephen, I just had a small question. I was struck by the fact that you see active actin comet formations in some of the punctate positions. And I was curious, what do you think those structures correspond to?
Yeah, that's a great observation. Yeah, so we noticed that too. So we've seen that in a number of mutants ranging from, so we saw it in the nervous rec mutant, the DAP160 mutant, and we also saw it in the Shabiri manipulation, in a different Shabiri manipulation. And so I think the idea, so one possibility is that these are just completely decoupled random assemblies. Another possibility is that they represent sort of a failure of maturation or scission of an initiated endocytic event, and this would be consistent with work from the DeCamilli lab. So he's shown both in synapses and in like macrophages, basically, that you can get this sort of comet-like actin assembly when you don't normally mature, and so I think that's maybe the most likely. Okay. So yeah, so now I'm going to tell you just again, so that stuff's all available online, and then this next part is stuff that's really all in progress, but I think is interesting to consider in terms of mechanisms of how we might control this sort of activity. And so again, kind of the challenge with understanding the molecular dynamics of these proteins is that they're basically everywhere, right? So we don't have these clean, distinct spots that we can track really clearly. And so to try to overcome that barrier and develop an approach to get some quantitative, more granular quantitative information about how these proteins are distributed at synapses, we developed this method where we look at one surface of the membrane, then we identify just local minima in intensity, so basically the dark spots within the mesh, and then we fill that in by water shedding and get this this segmentation where we can look at this meshwork and then ask questions such as, you know, how are these cells, or calling these cells, distributed across the synapse? What's the relative distribution of molecules between this meshwork and between the interior of the cell, that sort of thing. And so how can we use information like this? And so I'm going to just show you 3 slides of data on this that I think highlight some of the insights that we can get from this. So the first is that we can use this as a tool to map the relative protein concentrations, their regulatory interactions and relationships, and their dynamics. I think we can use this to quantify the relationships between the exocytic machinery, so active zones here, and the endocytic machinery here. And then finally, we can use this to look at the physical mechanisms that are able to assemble this, you know, kind of unique, interesting micron scale meshwork. And that includes protein-protein interactions, membrane composition, that sort of thing. And so just to kind of, again, I'll do 3 slides on those 3, 1 slide each on those 3 points. So first, we can see that the PAS proteins, which I think people have conceptually thought of as just being all looking the same, they're all in this endocytic zone. You know, the first thing that we notice is that there's really some interesting heterogeneity in how they're distributed within this endocytic meshwork. And so this is just examples of nervous rec in magenta and DAP160 in green, and then nervous rec and dynamin here in magenta and cyan. And so what you can see is if we quantify the ratio of the signal that's in the meshwork versus the amount that's sort of inside the cell, you can see that all 3 endocytic proteins, as you would very clearly expect, are all enriched in the meshwork, but not necessarily to the same extent. So dynamin, for instance, more of it actually intercalates inside the cell and is less restricted to the meshwork than these other 2, nervous rec and DAP160. And then just as a sort of a proof of concept that we're actually measuring something informative. If we compare this to an active zone component such as BRP, which is the homolog of ELKs, you can see that it's got the inverse enrichment. So instead it's enriched in the cell and not in the meshwork. So this is just a really simple kind of proof of concept that we're measuring what we think we're measuring, and that we can already start to see some interesting and unexpected differences in how these proteins are distributed. And so then we can use this type of measurement to ask how these different endocytic domains relate to the synaptic physiology using this active zone marker as a proxy. And so this really takes advantage of observations from the Littleton lab, where he showed that for each of these sites of vesicle release, the release probability scales with the intensity and size of the active zone, both in terms of the scaffold proteins like BRP and also the calcium channels. And so basically we can infer the release probability, the rate of exocytosis for each of these spots just from how bright it is. And so, for each of these data sets, we have this information, so we can look at, the intensity of the active zone and then ask, what's going on with the surrounding endocytic material. And again, I mean, you can see in the zoom how, dynamic here clearly is intercalating more into the zone, is coming closer to the active zone there. And so then to quantify that, I just looked at the Pearson's correlation between a number of different measurements and of the endocytic zone and how bright the BRP spot is. And so just as a sanity check, so the correlation between BRP and itself is negative with the mesh ratio. So the idea is that the brighter it is here, the less enriched in the mesh it is, which it is exactly what you'd expect, and positively correlates with how bright it is in this region. So so we're definitely measuring what we think we're measuring. And then when we look at the endocytic proteins, we see some interesting differences. So if we just focus on the mesh ratio, so the same measurement that I just presented a second ago, we can see that nervous rec, only nervous rec has a positive correlation with mesh ratio, but not DAP160 or Dynamin, but all of them have some positive correlation between the overall abundance and the active zone intensity. So suggesting that the amount of endocytic material is adapting to or scaling with the brightness of the active zone, so its propensity to release vesicles. And so then lastly, we can ask questions about what are the mechanisms that are regulating the formation of this reticulum. And so this is just comparing, again, the same control synapse on the left here, and this mutant which blocks the interaction between DAP160 and nervous rec. And what you can see is that the synapse on the right has a much more fragmented mesh. And that's quantified here with this mesh pore size. So basically, how big are all these gaps in the reticulum? And it's significantly smaller in the mutant. So suggesting that it's becoming fragmented and collapsed. And so the upshot here is that the same interactions that are controlling the activity of these proteins are also controlling their distribution and micron scale architecture. And so then just in the last little bit of the talk here, I just want to delve back into some biochemistry and ask what are some of the mechanisms by which these physical interactions might control this interesting micron scale organization of the endocytic architecture. And so this really takes advantage of the fact that we observe that if you just take some of these proteins, purified proteins, and mix them together, they form these liquid-like droplets. So many of you might be familiar with this from a number of different systems. Like virtually everything phase separates, and this is no exception. And there's a few reasons why we're particularly interested in this as either a potential mechanistic explanation of this reticulum, or at least as a model system to study its formation. And so some of the features here are that these endocytic proteins, both from our work and other people's work, have been shown to be prone to undergo liquid-liquid phase separation, and this is based on sort of the nature of the physical interaction between them. They're very weak and transient, and they have a lot of interconnectivity, which are essential features of protein interactions to drive this sort of physical effect. We know that endocytic proteins are really highly concentrated at synapses, and the formation of these liquid-liquid phase-separated condensates is a concentration-dependent phenomenon. So the higher the concentration, the more likely these are to form. And then finally, these can form these large micron-scale domains. And so it's just, since I'm going to focus on these for the next few slides, I just want to reiterate. So these dark droplets here, this is just concentrated protein in an aqueous buffer, and it's surrounded by more dilute protein also in an aqueous buffer. So there's no membrane here, there's no scaffold, there's no, it's literally just purified proteins and a salty buffer. And so what we think that these things can explain is really maybe potentially a lot about how these molecules behave at the synapses. So there's lots of examples in the literature now about how complex interactions among these sorts of liquid phase separated condensates can form interesting spatiotemporal patterns of organization. We know from other work that they can control vesicle clustering and traffic. And in particular, there's a number of studies now that have implicated this as a mechanism to control the extent and direction of actin assembly in a number of systems, including signaling clusters, but also during endocytosis. And so that's why we're really interested in this as a potential model system to study these. And so what I found so far is if we just take the protein-protein interactions, which can form these sort of dense interconnected meshes, protein interaction networks. So the first feature is that depending on the relative concentration, we can see really interesting differences in the condensates that form. So this is just sort of a titration of the stoichiometries of these different proteins. So each one of these panels is a different condition where the total protein concentration is 9 micromolar, but either with relatively more nervous rec down here, lower left, relatively more DAP160 on the right, or relatively more WASp as we go up. And what you can see is that as we add WASp, the amount, the brightness basically of DAP160, which is here in green, increases. So suggesting that we can tune the concentration of these proteins in the droplet depending on their relative stoichiometry. So the only important part here is simply that this is a controllable property that we can, even in these discrete spots, we can really control what's inside them. And then we can also control the spatial organization. So if we look at the droplets where there's both DAP160 and nervous rec, we see that nervous rec actually forms these much brighter, smaller internal puncta. So suggesting that there can be some complex organizations. And so we're still investigating the different protein interactions that are controlling all this, but the upshot is that these are tunable properties, and they can provide a really valuable system to understand how these behave at these very high concentrations. And we also know that they can control the material properties of these condensates. So I showed you that we can control what's inside them, but we can also control their physical properties. And so if you take a droplet that is just WASp and this DAP160 fragment, they behave very much like liquids. So as two droplets come together and fuse, they remodel rapidly into one larger round droplet. And so what we can do is track the kinetics of that and then get some insight into how viscous these droplets are. If we add nervous rec into this mix, we see now that the droplets still fuse, but now they remodel really, really slowly. So they have an almost 10-fold decrease in their speed with which they do this, suggesting that they're either much more viscous or have a much lower surface tension. And then, in thinking about what about nervous rec might be contributing to this effect, we considered that nervous rec has both these protein-protein interactions via its SH3 domains that I presented that control its activity, but it also can self-assemble and oligomerize via its bar domain. And so to determine which of these protein interactions might be contributing to this effect, I added a variant of nervous rec now that's missing its bar domain. So it can still interact with DAP160 and WASp, but it can no longer oligomerize itself. And this variant doesn't have this effect. So it no longer makes these droplets more viscous. So suggesting that this oligomerization effect might be important in controlling these physical properties. And just one last slide here. The same effects. So in thinking about how this might happen, why are these droplets more viscous? It turns out that unlike in liquid droplets where the molecules are typically rearranging on a very fast time scale, the nervous rec in these droplets is really not moving much at all. So if we frap a droplet here, and then we ask, how much does it recover over time? It really isn't recovering at all. So suggesting that the nervous rec molecules in these droplets are really, really stable in space. And so that might be one physical mechanism that could impose some level of order and really prevent the sort of remodeling that we see in droplets that lack nervous rec. And so again, just to kind of summarize that last part there. So the idea is that we can really use these endocytic molecules as a way to study phase separation. I think there's a lot of interesting features about the endocytic proteins in particular that could provide insight into this field, such as how very complex mixtures control the properties of these condensates, how specific membrane interactions control condensates, because we know membrane binding is an essential feature of these proteins. And then, how are the material properties of these controlled? And then we can also alternatively use this phase separation as an in vitro model system to study endocytosis in thinking about the localization and dynamics and organization of these proteins and also their activity. And then ultimately, I think by going back and forth between the in vivo and in vitro systems, we can get some insight into to what extent this is an actual mechanism to explain the in vivo behavior of these molecules. And so just to kind of put that in the context of the synapse, right? So the model here is that this periactive zone is this micron scale endocytic domain that, based on our new in vivo analysis, we think can adapt itself locally to the release properties of nearby active zones. And that these liquid condensates are really a great model system to study how endocytic proteins behave at these really high concentrations that we know are happening in vivo. And then the types of questions that we can ask are, how are these proteins, how are the pre-deployed endocytic proteins activated? So if we know that they're all at the membrane kind of hanging out, what are the switches, the molecular switches that are turning them on or driving them through these different phases of membrane remodeling? How does pre-deployment itself, how is it contributing to synaptic endocytosis, whether it's helping it be low latency, helping it be very scalable, high capacity, that sort of thing? How are endocytic proteins responding acutely to vesicle release? And then, whether there are any specific synaptic functions that are really uniquely require this large micron scale assembly. So I think these are all different questions that we can get at with this system. So I will stop there and say thank you to you guys for listening. Thank you to Avi for being a fantastic mentor, and to all the people who have just provided a great environment in the lab or contributed directly to this project.
Thank you, Steve, for that fantastic talk. And so now I'd like to just open the floor to questions from anyone in the attendees. Feel free to mute yourselves and turn on your camera.
So Steve, that was a wonderful talk. I love the kind of